Hello, and I hope everyone's doing well. We're going to take a look at Chapter 1, which is basically a general overview of what we'll be studying the year in AP Biology. This is a picture from our textbook, the 9th edition of Campbell Biology, and this is called a mother-of-pearl plant. And when you look at it, it kind of reminds me of plastic, really. And it makes me kind of wonder, is it even something that's alive since it looks so plasticky? Here's another shot of the mother of pearl, sometimes called the ghost flower. There I can see, it's looking a little bit more alive when you put it together with other members of its species. Think about that flower, think about how it's alive, and what characteristics make you think that it's alive. I want you to think back to your sophomore biology. How do you really know if something is alive? Do you remember that checklist? Keep thinking about that. By the end of the chapter, you should be able to answer that. And what would a scientist say about it? Also think about these questions. What's your present definition of science? And then, what's your present definition of biology? Then, how about this one? A headline reads, "A new cure for HIV is discovered." So, wow, that would be great news, right? But when you read, what would you look for in order to be able to have great confidence in this headline that it's actually true? By the end of this chapter, you should be able to answer that as well. As besides talking about biology, we'll talk about different aspects of the scientific method and different science process skills. This is a neat quote coming from our book right here. Biology is the study of life. It's rooted in the human spirit. People keep pets, nurture houseplants, invite avian or bird visitors with backyard birdhouses. They visit zoos and nature preserves. Biology is the scientific extension of this human tendency to feel connected to and curious about all forms of life. It's a science for adventurous minds. Yeah, I like to consider solve biological puzzles that once seemed unsolvable. The same discovery explosion that makes modern biology so exhilarating can also be intimidating. The key to understanding all that's happening is to recognize unifying themes that pervade all of biology. So hence, Chapter 1 is basically themes in biology. Different themes that we'll take a look at, such as science is actually a process, not just a big body of knowledge, but it's a process of inquiry that we go through, sometimes formalized, sometimes informal, but we go through that in our mind when we try to solve problems and learn more about our world around us. Evolution, the change over time, is a theme that pervades all of biology. Energy transfer, continuity and change, the relationship of structure to function. You might have heard the statement, structure determines function before. It definitely does. Regulation, the interdependence in nature, sometimes we also call that ecology. And then the relationship of science, technology, and society. By the time we're done with this chapter, we should be able to do the following. Briefly describe the unifying themes that characterize the biological sciences, distinguish among the three domains of life, and the eukaryotic kingdoms. Do you remember how many kingdoms there are? We should be able to distinguish between the following pairs of terms: discovery and hypothesis-based science, quantitative and qualitative data, inductive versus deductive reasoning, and science and technology. And finally, Since we'll be studying scientific method, identify the elements of proper scientific methods and be able to critique experiments. We'll practice that a bunch. Remember, life comes in many forms. It doesn't all just look like us. Remember, biology, the prefix bio means life, and logy is the study of. Science is a process. It looks different depending on what type of science it is you're studying, from being out in the field as a paleontologist to being in the greenhouse as a botanist, or possibly in the lab as a microbiologist or a geneticist. The processes that these scientists go through might look different, but deep down, they're going through some of the same steps that we would call the scientific method. And we'll be taking a look at that. Remember how it goes: making observations, asking questions, forming hypotheses, meaning a prediction as to what you think will happen, and then testing, which might involve an experiment or an observation, and finally making data and coming to conclusions based on that data. Your test may support your hypothesis, or it may not support it. The important thing is, as you're going through this method, that what you do is repeatable, so that you can tell this to other people, other scientists, other peers, and they can repeat what you've done, and they can also retest it. Your hypothesis must be able to be tested. The aspects of the scientific method are listed here. Biology usually follows the scientific method. It's usually peer-reviewed, published in scientific journals. Got to share that knowledge. Those results are repeatable, also open to change as we gain new data and evidence. After all, new technologies are created, so it's very safe to say that new data could be collected and therefore new evidence. So science is always open to that change. In fact, it's not good to say, or not politically correct to say, that you would prove your hypothesis. Basically, what I would rather you say is that you found evidence to support the hypothesis instead of prove it. Remember, a hypothesis must be testable and also falsifiable as well. There's a thing called the null hypothesis. When you were a sophomore, we didn't talk about this one, but we will now. The null hypothesis exists in an experiment, and it tells us that our variables will have no effect on the outcome. And we'll practice working with the null hypothesis much more in the future. But it's basically written to show that our hypothesis can be falsified. So let's say that I'm doing an experiment. Maybe I'm growing plants in a greenhouse, and I want to see what the effect of different colors of light will have on those growing plants. So possibly I have a white light. That would probably be my control, just regular plain old sunlight, white light. Then maybe I have just a green light, a red light, a blue light, a yellow light, and so on and so forth. So you might make a hypothesis that perhaps green light will make the plants grow the best. Then, going along with that experiment, you would have your null hypothesis, which would state that the color of light would have no effect on the growth of the plants. So, if that was true, then I would expect that all of my plants would be the same length after my experiment. One of the core themes in biology that we'll take a look at is evolution. The idea that all living things change over time. In fact, it's one of those characteristics of life. You probably recognize the name Charles Darwin as the father of evolution. Not that he came up with the idea of evolution itself, but rather the idea of natural selection. Organisms that are best adapted to their environment will have a greater likelihood of surviving and reproducing. This is such an important concept in biology that a famous scientist once wrote that nothing in biology makes sense except in the light of evolution. That's why sometimes it's so important to be able to understand natural selection and how that works. And then in understanding that, we understand how other ideas in biology work. Energy transfer. Life is an open system. Energy gets transferred through ecosystems. The ultimate source of energy on Earth is the sun, which then gives energy to our producers, the plants, and other photosynthetic organisms who go through the process of photosynthesis and make sugar, which then can basically feed the rest of us. And we get our energy from plants and other animals, and that energy continues to flow through our food webs. As it says here, entropy rules, as energy flows through our systems and is turned to heat energy, which unfortunately is a little less useful than other forms. Here's some good examples of energy utilization. Photosynthesis, a mosquito getting some blood, a cheetah eating a gazelle, and a koala eating some kind of plant. Continuity and change is another theme in biology. All living things reproduce and are able to pass on their traits to their offspring through DNA. Structure determines function. The structures that you have are shaped in a way for how they function, such as the bones in a bird. You take a look at them, they're very lightweight, very less dense than ours. And how about in the lower left picture, we have some neurons. Look at the way they're built for their job. It's being brain cells. They're transmitting information. And then in the bottom right corner, it's a mitochondria. If you look at the inside of a mitochondria, you see all these infolds of the membrane, which gives it more surface area, so it has more room to have molecules to make energy in the form of ATP for us. Structure definitely determines function. We're going to see that theme again and again as we look at the different aspects of biology. All living things need to maintain homeostasis, that dynamic balance of all their systems. Often they do this by some forms of feedback, known as positive and negative feedback. A very common example of negative feedback is how reproductive hormones are released in the body, such as our hypothalamus in our brain releases a hormone that goes to the pituitary, which then causes the pituitary to release a hormone that goes to either the testes or the ovaries, depending on if you're male or female. The testes release testosterone, the ovaries release estrogen. Those hormones then go back and shut off the hypothalamus from releasing more of that hormone that got the whole thing started in the first place. So those hormones, the testosterone and estrogen, act as basically an inhibitor to shut down the whole system. And why would you want to make more hormone and release more hormone if you already have it out there? Then you're just making more than you need. And once you use up the estrogen and testosterone, then there won't be anything holding back that feedback system anymore, so it'll reactivate and the hypothalamus will once again begin releasing hormones to get that whole system of feedback going. So that's what negative feedback is. It's basically a self-regulating process where some of the steps along the way inhibit the others so that you don't get an overproduction of a certain substance. Fewer things in the body are controlled by positive feedback. I can think of just a few offhand. Birth is one, as well as lactation, which is breastfeeding for new moms. Blood clotting is another one. And basically, it's like the release of a chemical, and it keeps releasing more and more and more. And then the only way to shut it down is something big. A professor that I had once called it a biological catastrophe, that you have to have a biological catastrophe to shut it down, such as in birth. You keep releasing the hormones that go to the uterus and make it contract, and then it gets worse and worse. The contractions get stronger and more painful until finally the end result is the baby's pushed out. I could make a joke there about a biological catastrophe, but I won't. So positive and negative feedback, ways of regulation or maintaining homeostasis in the body. So when I think of positive and negative feedback, I really think a lot about the endocrine and the nervous system with these. All living things maintain interdependence, meaning they can't live on Earth alone. They must have other organisms, whether it's just a simple example as feeding on other organisms like a food web. Or how about humans having bacteria in their guts in order to help you digest food and to make certain vitamins for you? Science, technology, and society. So science is the process that we go through to make new discoveries, to learn about the world around us, and technologies like the tools that we use in order to help us do that. We must do that, though, in the framework of society, so we must consider ethics, and sometimes we call that bioethics, in what we're doing. A good example of that might be cloning. Here's some other recurring principles and ideas as we go, that in life we all have definite diversity. Even looking at individuals in our own species, the humans, there's a lot of diversity in just the way we look, but yet there's this underlying unity that we share. So we have these things in common. Why do those similarities exist? Well, it all goes back to evolution. All things being connected through a common ancestor. And then branching off from there, called divergent evolution. Different changes over millions of years leading to what we see on Earth today. Due to natural selection and adapting to different environments. Creating from that common ancestor the different kingdoms of life. The regular bacteria, sometimes called eubacteria. The archaebacteria, which are actually more similar to us than the eubacteria. So we see that their branch is a little closer. The protists, then the plants, animals, and fungi. In order to be called living, you must exhibit certain characteristics. You must be organized by cells, at least have one of them, one cell. We're multi-celled organisms. You must be able to reproduce, and as we know now, all living things on Earth have DNA in order to pass their traits down. You must grow and develop, you utilize energy, you evolve and adapt to your environment, you maintain homeostasis, and you rely on other organisms. Life has a unique way that it's organized into different levels, from small to big levels, such as this is a molecule, our small level. Molecules build organelles. Organelles build cells. Here we have some plant cells. So back here, this is a chloroplast. So in this bigger picture, the chloroplasts are the green dots within the cell. These cells build tissues. So this is a plant tissue. That builds an organ. In a plant, a leaf is considered an organ, and then builds the whole organism, or the community if you want to go that route. Think to yourself, how would you list this with a human? Or would it be very similar? When studying life, some people prefer to go from small to big, taking the smaller items and watching how different properties emerge as you get to the bigger levels. This is one way to take a look at all living things. For example, a functioning bicycle emerges only when all of the necessary parts connect in the correct way. Or we can go the other way. The other way is called reductionism. Look at the big picture, such as the organism, and then work your way all the way down to the cell, and then even to the molecules that make up the cell, such as studying the DNA. Generally, in our studies, we'll take a look both ways, looking at emergent properties and also using reductionism. An important theme in life is that cells are our basic unit of life. All things are made of cells, or at least one. So cell theory was developed, especially when early microscopes were discovered. And today, at least, we know there's two basic types of cells that exist. There's the eukaryote cell. So that's this big one here with the definite nucleus inside and the different organelles that have membranes that close the organelle off from the cytoplasm or cytosol around it. And then this little guy here is the prokaryotic cell, just a really small thing compared to the big one. So I like to think of this, if I compared it, if the eukaryote cell was the size of a watermelon, then the prokaryote cell would maybe be the size of a raisin or a seed. Then there's a diversity of life. And then we divide that into these different characteristics. First of all, this is a newer division, dividing all forms of life into domains. They didn't have this when I went to college, but they do now. So all living things can be put into these three categories. They're either the regular bacteria, sometimes called eubacteria, archaea, or eukarya. Funny thing is, not only do these two, bacteria and archaea, get their own domain, they also get their own kingdom. So both bacteria and archaea have prokaryote cells. The rest we name after the eukaryote cell, eukarya. And so these four kingdoms, Protista, Plantae, Fungi, and Animalia, all have the eukaryote form of cells, meaning they have a nucleus with membrane-bound organelles. So the division of life, that's called classification or taxonomy. And we can take it a step further. We can go further down. Domain would have been here at the bottom. Then kingdom, phylum, class, order, family, genus, and species. Remember, the genus and species is what makes up the scientific name. The taxonomy is an attempt to bring order and unity to the diversity of life. So, why study the themes of biology? Well, biology is an ever-expanding body of knowledge. There's so much. Just look at our own textbook. It's huge. It's too much to memorize it all. So we need to generalize and create this framework so that we can organize our new knowledge. And what part of biology is growing fastest? I would say it's probably the genetics part. We've made huge advances in genetics. You've probably heard of things like cloning and stem cell research, the human genome, being able to subscribe to sites like 23andMe and Ancestry.com and sending in your DNA and getting a sequence back within a small amount of time. The first time we figured out a human sequence, it took 10 years. Now we can do it in a matter of hours. This is a huge growing field. And then the development of CRISPR technology, which means we can edit our own genes. Let's take another deeper look at the scientific method. Did you know that the word science is actually derived from Latin? It means to know. It's a process that we go through to discover things about the world around us. It includes observations, making hypotheses, and then testing them. When we get data from our tests, basically we can put that into two different characteristics: quantitative data and qualitative data. Quantitative data generally has numbers. Qualitative data is kind of now being recognized as good data as well, but would entail more observations and descriptions, like what Jane Goodall did in her studies of chimpanzees. This is her actual journal of tons of observations that taught us a lot about this species and close relative of ours. Scientific method often uses inductive reasoning, where you would draw conclusions. Basically, you repeat specific observations over time, and then you are led to a generalization, such as in my own classroom, I look out the window every morning and I see the sun come up in the east windows. That happens every morning. So therefore, because of that observation that I've repeated again and again, I can make that generalization of inductive reasoning that the sun always rises in the east. Observations and inductive reasoning lead us to ask questions, and then those questions can lead us to hypotheses. A hypothesis is a tentative answer to a well-framed question. Then it leads to predictions that can be tested through observation or more formal experimentation. For example, here's a classic. Your flashlight doesn't work, so you ask yourself, why doesn't the flashlight work? You might make a few hypotheses in your head, such as the batteries are dead or the bulb is burnt out. Both of those are actual testable hypotheses. So here we go through the steps of testing each of those hypotheses. In the first case, when we check the dead batteries and we replace them, it still doesn't work. So what we would say there is we falsified our hypothesis that it was dead batteries. So often what happens is then we go back to the beginning, go back to the question, make another hypothesis, such as it's a burnt out bulb. Then we replace it, test it again, and sure enough, now the flashlight works. All right. Remember I said earlier, don't say that you proved your hypothesis there. You just have evidence to support it. Evidence to support your hypothesis. Or you did not falsify the hypothesis. Here's a great example of why I should never say I proved my hypothesis. For example, let's say we replace the bulb and it now works. Sure, it supports the hypothesis that our bulb was burnt out, but it doesn't prove it, because what if by chance the bulb wasn't even in there correctly in the first place? That's a thinker, huh? And in science, we want to make sure our hypothesis is testable and falsifiable. So a hypothesis like a ghost fooled with the flashlight, and that's why it didn't work, we don't have the ability to test that, at least not yet, in science. So supernatural and religious explanations are outside the bounds of science. The process I just showed you is an idealized process. Usually scientists don't go through every step in such a formalized manner. It would be ideal if they could, but usually it's not like that. Now let's talk variables. In an experiment you have variables, and hopefully you remember these words from past classes: the independent variable and the dependent variable. We'll be working with these a lot in the next few days. When we're doing an experiment, we should be able to identify the independent variable as the one that's manipulated or changed. Such as I mentioned an experiment earlier in my lecture about planting plants in a greenhouse and seeing which color of light makes them grow best. So in that case, my independent variable would be the color of light. My dependent variable is what we measured. This is where we get our data from. So the actual measurements that I take, probably the growth of the plants, that would be my dependent variable. So remember, independent variable is manipulated or changed. Dependent variable depends on the independent variable, therefore it's what we measure. In that experiment, we would also need a control group. This is where the independent variable is omitted or left out, or sometimes just held at an established level called a standard. In my plant experiment, white light would be the control group, or just plain sunlight. Then there's other controlled variables that we want to maintain at a constant. These controlled variables have the ability to become their own little independent variables if you let them, so you want to control them. So some examples might be that I would give all my plants the same amount of water, the same amount of fertilizer. I'd have them in the same environment, the same amount of light each day. I would want to control everything I possibly could so that I could be very sure that the independent variable is the one affecting the experiment. So now that I've finished my brief overview of Chapter 1, let's see if we can answer these questions. Number 1, what's the difference between inductive and deductive reasoning? Remember, inductive reasoning is where you make a lot of observations over time, and then you make a generalization based on that observation. I just realized I forgot to include the slide for deductive reasoning, so I'm going to go back to that. Here we go. Here's the deductive reasoning slide. I skipped over that one. Sorry. This uses general premises to make specific predictions. It's basically recognizable if you have an if-then statement. And it's basically how you would want to write a hypothesis if you could. If you could write a nice formal hypothesis, you would write it in the form of deductive reasoning and have an if-then statement, such as the one here. If organisms are made of cells, and if humans are organisms, then humans are composed of cells. Or you could transfer that to when I did my plant experiment. How about this one? If plants reflect green light, then if I put them under a green light, they won't grow. Or they'll grow less than if I put them under other colors of light. There we go. So there's our difference between inductive and deductive reasoning. the elements of a proper controlled experiment. Remember, that's a formal experiment like this, with observations, a question, a hypothesis, prediction, testing it, and then conclusion based on the data. Remember, it's not always that idealized. In an ideal world, it would be. Let's put these into a hierarchical order from small to large. If you'd like to pause the video and do it yourself, do that now. And now I'm going to give you the answer. The smallest thing there is a protein. That's just a molecule. Then a cell. Then the smooth muscle tissue. Then an organ level, a small intestine. Then we'll go, let's see here, system level, digestive system. Then a system would build a student. Then all the students make up the Albertley Tigers, and the Albertley Tigers are in southern Minnesota. Ooh, write the equation for photosynthesis. Remember how that works? That's an example of energy flow in an ecosystem. The fact that plants pull carbon dioxide out of the air, and they use water and sunlight, and then they produce sugar, usually in the form of glucose, and then also oxygen. And describe where Earth's energy ultimately comes from and how it gets to us. Well, that would be from photosynthesis. The ultimate source of energy on the Earth is the sun, and plants transfer that energy to us through the process of photosynthesis, making sugar, storing that, and then giving us and other organisms something to eat. Alright, this is a long one. If you'd like to pause the video and read through that, that is a great example of positive feedback. That's ovulation, what actually happens. And then question 7, if you'd like to pause the video and read through that one, that's an example of negative feedback. In fact, it's the example I gave you earlier. Finally, I'll go back to our learning targets for the chapter, read through those, make sure you can do those things. Again, we'll practice some of the things I talked about in the short lecture here. It's also a short chapter. I encourage you to open up the book and read through that. We'll go back to these questions. How do you know if something's alive? Make sure you know what those unifying principles of life are as well. A definition for science and biology, and what you would look for in a scientific study. All right, any questions? Make sure you let me know.